Hi everyone, this video is to go over DNA mutations practice worksheet. And this worksheet's really important because sometimes mutations can seem tricky at first, but there's a few things I want to go over about mutations. First of all, sometimes they're neutral, they don't affect us at all. Sometimes they're beneficial, like with the Teenage Mutant Ninja Turtles, or in terms of helping us evolve. And sometimes they can be harmful, like in terms of cancer, okay? When we are looking at this worksheet, this top box represents the original DNA sequence. What is it supposed to be? Okay. And then down here, we've got a mutated DNA sequence and another mutated DNA sequence. Something that's really important to understand is that when we are working through these, you always want to code the amino acid based on the mRNA. So the very first thing we're going to do is we're going to look at this DNA strand, and it's already separated into codons for us, or those three bases. We are going to transcribe it in the nucleus to get mRNA. Then, once mRNA has been transcribed, that's when we can actually figure out our amino acids using our codon charts and put those together as monomers to create a protein. Okay, so another thing I want to go over is when we are transcribing from DNA to RNA, it's a little bit different than DNA to DNA and DNA replication. In terms of DNA replication, A always pairs with T, apple in the tree, and C always pairs with G, chips in the guac. However, with RNA, any type of RNA, A is going to pair with U. So the way I remember that is aunts and uncles. And C always pairs with G because chips and guac never lie, all right? So the very first thing we're going to do is we're going to take this DNA sequence and transcribe it into mRNA. So to start off, TAC. T pairs with A pairs with U, and C pairs with G. A great way to remember what amino acid goes with AUG is school always starts in August. So AUG is actually the start codon. That's really important when we're looking at these strands. Let's go on to the next one. CAA. C pairs with G, A pairs with U, and A pairs with U. Moving on. AAC. A pairs with U, A pairs with U, and C pairs with G. Next one, CGC. G, C, G. Second to last one, TGC. T pairs with A, G pairs with C, and C pairs with G. And last but not least, TGA. T pairs with A, G pairs with C, and A pairs with U. Okay, now we went ahead and we've transcribed DNA to mRNA in the nucleus. Now we need to figure out what amino acid does that mRNA codon represent. When you are using these codon charts, always, always, always use the mRNA strand. Oftentimes, students will make the mistake of using tRNA. tRNA carries the amino acid, but mRNA is what codes for it. It's what makes the tRNA key come to that lock, okay? So the next thing we're going to do is look at the amino acids, okay? There are two different charts you can utilize for this. I don't care which one you use. However, I do want you to be able to be familiar with both. So in this one, it's a circle, and you're going to always work from the inside out. On this one, it's a chart. This is the one that I grew up learning how to do, and it's going to be a lot like a multiplication table. So you're going to start with first base, second base, and third base. We'll go over that shortly. Let's go ahead and start with this round one. So the very first one is AUG. You should memorize that. School starts in August. That's a great way to see, am I using these codon charts correctly? So when we're looking at this, A, then go to U, then go to G, and you're going to see that it says methionine. Methionine is the start codon. Let's go ahead and do the other one now. AUG. Always start right here where it says first base, then second base, then third base. Let's go together. A, then follow to the U up top. You can notice that U is right here. Okay, so AU. And then you look for G. AUG is met or start. Okay, so the very first amino acid we have here is met, or start. GUU. Let's start with this one first. GUU. So we're going to start with G, we're going to go to U, and then U. That's going to be VAL. Notice how GUU, GUC, GUA, and GUG all code for vowel. That's very important. GUU codes for vowel. UUG. Let's use the other one this time. UUG. UUG. Leucine, LEU. LEU. GCG. Let's use this one now. G, C, G is going to be ALA. ALA. ACG. Let's use that round one now. ACG. A, C, G, THR is going to be that one. THR. And ACU, A, then C, then U, same exact one. Now, let's look right here. A, then C, then U, THR again. That's a great way to double check. Okay, so that is our original DNA strand. You have taken DNA, you've transcribed it into mRNA, and then you've translated that mRNA into amino acids, which make a protein. Now, let's talk mutations. Somewhere between this original DNA sequence right here and this DNA sequence right here, there was a change. We need to find that first, okay? So we're going to be comparing the mRNA to the mRNA and the amino acids to the amino acids. So TAC. TAC is the exact same thing as above. So that's going to be AUG, okay? Hasn't changed yet. CAG in the mutation, but CAA up top. Ooh, we got to change here. CAG would be GUC. Okay, so that's different. Let's go ahead and bold that so we don't lose that, or underline it. All right, AAC, that's the exact same as in the original DNA sequence. That's going to be UUG. No change there. CGC, that didn't change from the original DNA sequence. C pairs with G, G pairs with C, and C pairs with G. TGC, didn't change from the original DNA sequence, as you can see here. ACG. Next one, TGA. TGA did not change. So that's going to be ACU. Before we even look at the amino acids, did we change the entire DNA strand? No, because that first codon and all of the succeeding codons are all the same. However, this one is different. Instead of having CAG, instead of having CAA, we have CAG. So we replaced the A with a G. We substituted it. We substituted that. So what type of mutation is this? Even before doing the amino acid? Substitution. That's it. Okay, we substitute that. Now, we don't know if this altered just one or all of them, or do we? AUG is going to be the same. UUG is going to be the same. CGC is going to be the same. All of these are going to be the same, but is GUC any different? Let's go ahead and go through. We know all of these other ones are the exact same, so we don't really need to fill out the entire amino acid sequence. But GUC, is that different than VAL? Let's check it out. GUC. GUC. That's VAL. Ooh. So, if VAL is for GUC and GUU is also VAL, did we change the strand at all? No, it didn't change at all. Even though the DNA sequence, there was a substitution, there was absolutely no change at all. This is really important. At the very beginning of this worksheet, I told you that there were 3 ways that mutations can change our DNA. Neutral, where it changes it, but ultimately the protein ends up the exact same way, which is what happened here. It could be negative and change the entire thing, or it could be beneficial and change the entire thing. So there we go there. That's a substitution because we substituted the C for the U. All right, moving on. Now, this looks different. Let's compare and contrast. TAC is the same. CAA is the same. AAC is the same. CGC, ooh, that doesn't look the same. All right. TGC, ooh, that doesn't look the same. And then TGA, that doesn't look the same. This is going to be a lot different than the first one. So let's go ahead and go through the entire mRNA sequence. TAC, well, T would pair with A would pair with U, and C would pair with G. All right, we got the very first one. That's the start codon, or methionine, or MET. Go to the next one. CAA. CAA is the same up here, so we're good so far. GUU. Now, AAC is going to be the same. A pairs with U, C pairs with G, and C pairs with G. But here on out, it's going to look different. So let's make sure to underline those so we keep track. GCG is now going to be CGA. GCT, same thing, CGA. And last but not least, GA is CU. Okay, so these first three are the same. This one's different. This one's different. And this one's different. And remember, we're comparing this mutated mRNA and DNA sequence to the original DNA sequence, okay? So let's talk here. Was this a substitution? Did we just plug one in and take that exact one out? No. It was either an addition or deletion, okay? So let's clear that up. One great way to figure this out is, do you see how this doesn't have a third base? It says GA, and this one had an end of 3. We know that it's missing one this way. It's a deletion. Now we need to find out where that's at, okay? Oop, deletion, because we have one less. Now, let's say we had all 3 of these, so GA and then another letter and then an additional one. That would be an addition. We plugged something in. So what we have here is somewhere in this code, we deleted one of those bases and it shifted everything over. Here, we plugged one in where the other was taken out. So only that particular amino acid was changed. In this one, it changed all of the other things from that point. So let's talk about where this happened. We had ACC the same as up here. So we know it's going to be in CGC. CGC. Ooh, the C was taken out right here because then right here we had TGC. So all of those are shifted over. So right before this G, there was a letter there. There was a part of a DNA strand. So how did this affect it? Well, let's start off from the beginning. We know AUG is the same. We know GUU is the same. We know UGG is the same. So let's go ahead and start at CGA. On this one, we should have had ALA, okay, for the amino acid. However, we have a mutation here. So let's go ahead and start. CGA. Let's use this one. C, and then G, and then A, we have ARG. Ooh, okay. So that's a big difference. It's a completely different amino acid. CGA. In this top one, it would have been THR. Let's check it out. CGA. C, then G, then A, ARG again. That's different from there. And then this one was THR again. Let's check this one out. CU. CU. Oh, we don't have a third one. So actually, that doesn't even equal an amino acid. Okay? So what changed here? No change? Something definitely changed. Only one amino acid changed? No, we had 3 change. Premature stop signal? No, we didn't hit a stop signal. No stop signal? Yeah. one amino acid added or deleted, or all of the amino acids change after the point mutation? Well, there isn't a stop codon, but the big thing that's changing here is all of the amino acids are changed from that point forward. Sorry, guys, the dictionary always wants to pop up. So the differences between this second mutation and this top mutation are here, we substituted it. We only replaced one letter. Just like a substitute teacher, your whole day isn't disrupted when Mrs. Mormon and Mrs. Duffy or any of your teachers aren't there. Just that period is different. In this one, imagine that one of those classes was completely deleted from your schedule. All of your other classes would be shifted forward. This is a point mutation, substitution. This is a frame shift mutation because you're going to shift your entire DNA sequence from that point. I hope that clears some things up. Please make sure to reach out to your teacher for any extra help.